So how the question is going to be addressed on the lab exam? So first, you're going to have to know the identification of the tissue. So it's going to be mainly based on diagrams. All right, so I'm sorry for this. So the the lab exam is going to be diagram based. Maybe you're going to have slides like those ones on here. And the best way for you to be able to identify the tissue is to follow the pattern of identification that we have been following in order to make sure you get the correct answer. So on the exam, you're going to have a list of choices. This might be around 20 to 30 choices that you're picking the single best answer from for each of those uh, tissues. So you're going to need to be able to identify the tissue. You're going to need to be able to know the location, the different locations where this tissue is going to be. And you need to be able to state the different functions of this tissue. So a quick review of what we've been discussing. If you remember, we've discussed the epithelium. First, we've got 4 types of tissues. We've got the epithelium, connective tissue, muscle tissue, and nerve tissue. So if I'm looking at a slide, very first thing, I need to know whether I'm looking at epithelium or connective or muscle or nervous tissue. So in order for me to identify, to make sure that I'm looking at an epithelial tissue, so I need to find a space, and this space is lined with cells. It might be a small space lined with cells. It may be a space outside of an organ, so it's going to be large, but 2 components here for me to identify that the tissue I'm looking at is an epithelium. I need to find space, a clear space, an empty space, and it needs to be lined with cells. So space, we can all agree on how to find the space. It's empty, it doesn't have anything in it. And it's lined means it's going to be surrounded by the cells. And what I mean by cells, those are living cells. And in order for me to identify a cell on a slide, I need one of 2 things. Either I can see boundaries, or I can see the nuclei, or both of them. So in order to say that I'm looking at cells, and this is very important, cells, many cells, I need to find either the boundaries or the nuclei or both of them. So how will I be able to classify the different types of epithelium? So now I figured out that this is epithelium. I found space, and this space was lined with cells, many cells, not just one cell. So how would I be able to identify the epithelial tissue type? 2 factors. You need to look at the number of cell layers. You need to look at the the shape of the apical cells. So if I have one cell layer and those cells forming my layer are flat, so we're going to classify this as simple because I'm formed of one cell layer, and we call this epithelium that is formed of flat cells, it's squamous. So this is going to be simple squamous epithelium. If my epithelium is formed of one layer of cells, but those cells are cube-shaped cells, so I'm looking at simple because I'm formed of one cell layer. And if I'm looking at cube-shaped cells, this is going to be a cuboidal epithelium. If I'm looking at one cell layer, which cells are tall, so those are going to be forming my simple columnar epithelium. How to figure out the shape of the cell? We have two ways. Either you check the height compared to the width of the cell. If the width of the cell is greater than the height of the cell, it's going to be a squamous cell or flat cell. If the height is almost equal to the width of the cell, this is going to be a cuboidal cell. If the height is much greater than the width of the cell, this means I'm looking at a tall cell, so it's going to be a columnar cell. Another way for us, if we can't see the border, so we are using the other method only if we can't see the border, which is going to be the shape of the nucleus. The nucleus flattened like this, ovoid, squeezed. So this is going to be inside a squamous cell. If the nucleus is rounded, it's going to be a cuboidal cell. If it's ovoid but elongated, like this is going to be a columnar cell. So again, how do I figure out the shape of the cells that I'm looking at? You have two things. The very first method, this is the main method, and we're going to use it if we can't see clear borders of the cell. I can't see the borders of the cell. I can see the membrane of the cell. I can see the boundaries of the cell. So with this into consideration, I will measure the height and compare it to the width of the cell. If the width of the cell is greater than the height of the cell, it's a squamous cell. If the height of the cell is equal to the width of the cell, it's going to be cuboidal cell. If the height of the cell is greater than the width of the cell, so this is going to be a tall cell, it's going to be a columnar epithelium. All right, everybody? Easy so far. So how would I know whether I'm looking at one cell layer or more than one cell layer? You need to have the cells forming more than one cell layer on top of one another. So I have one basement membrane, and on top of this basement membrane, I got more than one layer of cells stacked on top of it. So in this case, I will name it a stratified epithelium. Stratified epithelium. All right, so again, which cell of those layers would I be identifying the shape of? Look at the top cell layer, the apical cells. Apical cells. So if you remember, here I can see the boundaries, so I will check the height and compare it to the width of the cell. So it's much wider than long. So we can consider this as a squamous cell. So what am I looking at in here is going to be a stratified squamous epithelium. But how come I do see here columnar cells? We're not concerned about the basal cells or the middle cells. What we're concerned with to identify the type of epithelium is going to be the top cells, the apical cells. Yeah, they need to be at the top. They need to be at the top of the tissue and they need to be cells. What if I do have fibers here? Thick layer of fibers. Don't let this confuse you. Why? Because those are not cells to consider. You're gonna see what is the last layer that still have nuclei in it. And this is gonna be your apical cells. Although it's not the top layer in this tissue, although it's not the top layer in this tissue, but it is the top cell layer. Is the difference clear? All right, so stratified squamous epithelium might be either without those dead cells on top of them. Those is my keratin layer. So it's going to be non-keratinized. Or it has keratin layer on top of the top cells. So this is going to be keratinized. What if I've seen a type of tissue that looks stratified? I can't see many cell layers on top of one another. But those top cell layer, this top cell layer, when I look at its cells, I see that the cells are columnar. And when I look at the basal cell layers, I see they are much smaller in size. So I look here, I'm about to say it's stratified. It's stratified, many cell layers on top of one another. But when I looked on here, I found that the cells at the base are smaller in size compared to the cells at the top. Much smaller in size. So this doesn't make sense here. This doesn't make sense. Why? Because remember, the epithelium was avascular. It gets its nutrients and oxygen by diffusion from the underlying connective tissue. So there is no way that those cells close to the source of nutrients and oxygen are smaller in size compared to the ones at the top, which are supposed, I'm supposing that they are getting less nutrients and oxygen. So when we looked closer to this tissue, we find out that all the cells are actually attached to the same basement membrane. So how many layers of cells do I have? I have one cell layer. I have just one cell layer. All of the cells are attached to the basement membrane. All right, so for example, here, if somebody is not convinced, so in my front yard, I've got a palm, and a tree coming out. Just behind So when I'm looking from this side, this is me standing in front of the palm, and again, there is a tree coming out just behind it. So what appears to me? What will appear to me is the tree on here. If I'm looking from an anterior view, not a side view, we'll see the branches of the tree. Like this. And below it, you've got the palm. So how many levels do I have? How many levels? For the first impression, when I see palm, and on top of it, I've got tree. Are those forming the same layer? Are both attached to the ground? Yes. So I have one layer of trees, right? In my front yard, I have one layer of trees. But when I look from an anterior view, this is what looks like from an anterior view. I see a palm, and on top of it, I've got the tree. This is the same concept on here. This is the same exact concept. So why do I get this false impression that I've got one tree and on top of it I've got another tree? Because they are so close to one another. They are crowded against one another. So when I look from this anterior view, I see two layers of trees, as if they are growing on top of one another. But actually they are not growing on top of one another. They are all grown from the ground, but one is taller than the other, and they are so crowded, so I get the impression that I've got one layer, and on top of it, I've got another layer. Does this make sense? All right, so for the first impression about, and I'm about to say it's stratified, but when I've seen that the top cells are columnar, I can just think of it in another way. So even if you can't see that all the cells are attached to the basement membrane here, if you told me I can't see that all the cells are attached to the basement membrane, neither I am. But it's a matter of logic here. It's a matter of logic. This is what you're going to rely on to know this type of epithelium. It's a matter of logic here. You can't have small cells and on top of them, you're going to have larger cells. Because as you keep going up, you get less nutrients, less oxygen. So this is going to be a false stratified, a fake stratified. It's what we call pseudo-stratified. And the top cells, apical cells, are columnar. So we're looking here at pseudo-stratified columnar ciliated epithelium. One last type of epithelium that we've seen, it was formed by more than one cell layer. Some of the apical cells were cuboidal. And how did I figure out that those are cuboidal? I check the width, the height of the cell and compare it to the width of the cell. They are almost equal. And see on here, another cell, it's much wider than it is long. So some cells are cuboidal, some cells are squamous. But you notice something very important, that those squamous cells are still large compared to those ones. Those are squamous, those are flat, because they got less nutrients, less oxygen by diffusion. But those ones are still large in size, but they are just flat shaped. So they didn't lose the oxygen, they didn't lose the nutrients. That's why they are squamous. What happened to them is stretch. So here we're looking at a type of epithelium that has the ability to change the shape of its cells. So why do I need to change the shape of my cells? To accommodate stretch. And this is the type of epithelium that's going to be located in your urinary tract, your ureters, urinary bladder, and urethra. This is, so we decided to not classify it in a regular way, stratified simple. We relied on the fact that it changes the shape of its cells, so we called it a transitional epithelium. Something also to pay attention to here when we are identifying the epithelium, that simple columnar epithelium has two varieties. Either the cells have cilia, so we're going to call this as a ciliated simple columnar, or they don't have cilia, so we're going to call those as a non ciliated simple columnar epithelium. It's either ciliated or non-ciliated. The same for the pseudostratified, because the pseudostratified is just, it's simply simple columnar, but the cells are so crowded against one another. I have lots of crowded cells, but it's essentially the same like simple columnar. I have one row of cuboidal cells, but the cells are so crowded. This what gave me the impression that I'm looking at more than one cell layer. So I still have two varieties here of the pseudostratified columnar, similar to what I've got for the simple columnar. So it's either going to be non-ciliated if the cells do not have those hair-like projections, which will allow the propulsion of the mucus, or they do have, so they would be ciliated. So they might not have cilia, so we call them we call this type of epithelium a non-ciliated, or we will have cilia on top of the cells, so we're gonna classify this as being ciliated. All right, so again, how did I know that this is epithelium in the first place? You need to see that you've got a space that you are lining with those cells. This might be a lumen of an organ. This space might be a lumen inside an organ, inside the tube, or the outside, if we're dealing with outside of the organ or outside of the body even, if we're looking at the skin and the covering of some mucus membranes. Any questions here about what we've stated so far? Any questions? All right, great. So moving on to the connective tissue. So a very simple way to make sure that you're looking at the connective tissue is by excluding the other types of tissues. So it's not epithelium because I don't have a space lined with cells. It's not nervous because I don't see the nerve cells or the neurons, which have a very characteristic shape. Cells are going to be present with processes coming out. Many processes are coming out of the cell, and in the background I would see other sets of cells. Those were the supporting cells. I see the nuclei of the other cells in the background like this. And it's not a muscle tissue. Why it's not a muscle tissue? Because a muscle tissue is going to also be formed of cells. Throughout the tissue, I will have cells. Those cells might be either cylindrical shaped cells. This might get confused with the connective tissue because they will look like fibers, but again, those will have nuclei embedded inside the cell. That's why I can't consider them fibers. I can't say those are fibers. Those are living cells, and even each one of them has more than one nucleus. Those are cylindrical shaped cells. They have dark lines, light bands, dark bands, light bands, dark bands, light bands. Those are what we call the striations. This is a very characteristic appearance of a special type of muscle tissues. This is my skeletal muscle tissue. might have cells. How did I know that those are cells? They have clear boundaries. They might show some nuclei. In some cases, you won't see nuclei. They are cylindrical shaped cells. They are still striated. They have dark lines throughout the length of the cylinder. And I will have even darker lines connecting the adjacent muscle cells. This is a very important characteristic that we can use to identify this type of muscle tissues from the skeletal muscle tissue. Also, another main characteristic of this is my cardiac muscle tissue on here, is that its cells are branching. Unlike what we've seen here in the skeletal muscle tissue, cells were just cylindrical in shape, they are not branching. So two factors on here, if you're seeing cells that occupy the whole thickness of the slide, of the tissue, and this is not a type of cells that will form the coverings and linings of the organs. Those are not epithelial cells. So I don't see a space lined with cells. So I'm occupying the whole thickness of the tissue by myself. So this is going to be a muscle tissue. If the muscle tissue is formed of cylindrical shaped cells, they are striated, multinucleated, those are skeletal muscle cells, and this is going to be my skeletal muscle tissue. If I see cylindrical shaped muscle cells, they are also striated, but they do have those dark bands connecting the adjacent muscle cells, and the cells are branching. So those are the cardiac muscle cells, and this is going to be my cardiac muscle tissue. Easy, easy. What if I'm looking at cells occupying the whole thickness of the tissue? Again, I'm not lining a space. I'm not nerve cells. And those cells are spindle-shaped cells. Spindle-shaped cells. Each of them has one nucleus. They are mononucleated. And there is no striations. I don't see any striations on here. So what kind of muscle tissue are we looking at in here? We're looking at smooth muscle tissue. Smooth muscle tissue. All right, so if I'm looking at a slide and the tissues that I'm looking at, trying to identify, it's not epithelium, it's not, doesn't have a space that is lined with cells, so it's not epithelium, it's not formed of nerve cells, so it's not going to be a nervous tissue with those cells having processes coming out. And I'm not formed of cells occupying the whole thickness of the tissue. They are either cylindrical shaped, multinucleated, and striated skeletal, or they are striated cylindrical, but they are branching with intercalated discs in between the cardiac muscles, the cells. So this is going to be a cardiac muscle tissue, and it's not formed of spindle-shaped cells, which are not striated, and they are mononucleated. So because this is going to be my smooth muscle tissue. If I rule out all this, so what remains here is going to be connective tissue. Connective tissue. One important characteristics that I can tell you to look for when you are looking at a connective tissue is the presence of connective tissue fibers. And what differentiates the fiber of living cell is going to be the presence of nuclei. So if I see just fibers, those are protein filaments like this. They are in multiple shapes and forms. But if I see them, this is going to be definitely a connective tissue. This is going to definitely be a connective tissue if I see connective tissue fibers. And the three common forms of connective tissue fibers are those thick bundles of fibers that we call collagen fibers, or those thin, darkly stained long fibers. Those are what we call the elastic fibers. And we've got short branching fibers. Those are called my reticular fibers. So this is going to be one of the main things that I will rely on in the identification of the different types of connective tissues. The type of fibers present. So if I see fibers, this is going to definitely be a connective tissue. Two types of connective tissues that do not have clear fibers in them. For us to say that this is a type of connective tissue once we looked at, but thankfully they do have very characteristic shapes. So all of the different connective tissues that we've seen did have some connective tissue fibers, but remember, we've got two of them that didn't show much fibers in their structure. We couldn't see those fibers. In my opinion, those are the blood tissue, which is a type of connective tissues. Very characteristic appearance. I have a clear matrix, which is my plasma. It's clear. It has nothing in it. What I can see scattered on this clear matrix are the different types of the blood cells. And the most common of them, it's going to be the erythrocytes or the red blood cells. The erythrocytes will have very characteristic appearance. They are reddish in color, with dark peripheral edge and pale center. So I'm looking here at the erythrocytes. Or what we call the RBCs, or the red blood cells, or other textbooks will call them red blood corpuscles because they don't have a nucleus. They are enucleated. They don't have any nucleus in them, so we don't call them cells. A better name for them, it's going to be corpuscles. Other cells that we might see are the white blood cells or leukocytes. Leuko means white and cyte is cell. So leukocytes are my white blood cells. And the white blood cells, the two most common forms that I will see in the blood are the neutrophils, which I will see an undefined shape of a nucleus inside with lots of scattered granules in the cytoplasm. Those are my neutrophils. And again, neutrophils are a type of leukocytes. Another very common type of white blood cells, it's going to be a cell with a dark large nucleus that occupies most of the size of the cell. This is a highly active immune cell. This is the cell that we're going to be trying to activate types of by vaccine. This is a type of cells that will be affected by HIV infection. So this is one of the most active immune cells that we do have in the body. They play a major role in fighting viral infections, bacterial infections, cancer cells. We call those are my lymphocytes. Another type of connective tissues that I don't see clear fibers because the fibers got squeezed between its cells. The cells are occupying most of the size of the tissue. They look large in size. They are vacuolated, large vacuole inside. And those large vacuoles were filled with triglyceride or fat. Those large vacuoles were compressing the nucleus against the wall of the cell, which gives me this signet ring appearance of the cell. Those are my fat cells. And why I don't see fibers, this doesn't mean I don't have fibers in my structure, but the fibers are simply squeezed away by the effect of the enlargement of the fat cells. All right, so you still have fibers, but in most cases you won't be able to see them clearly. So this is going to be my adipose connective tissue. So you can see on here, Adipose connective tissue, I don't see clear fibers. Blood tissue also, I can't see clear fibers. Another type of connective tissues that has a very characteristic appearance, it's going to be the bone tissue or the osseous tissue. The bone tissue is formed by building blocks. We call those the osteons. Those osteons are going to be formed by concentric layers surrounding a central canal. And the central canal is where the blood vessel is going to be traveling. We call this building unit of the osseous tissue or the bone tissue is my osteon. The dark canal here at the center is my central canal. And surrounding the central canal, I have layers of calcified protein. And those layers of calcified protein on here are what we call the lamellae. Embedded in those lamellae, we've got tiny little spaces where the mature bone cells are going to be kept. We call the mature bone cells on here. Those are the osteocytes. And those spaces, tiny little spaces that I have, those are my lacunae. So we're looking here at osteocytes in lacunae. So we see on here. This is my blood tissue. Clear background. Erythrocytes are going to be the most common type of blood cells that you can see on the slide. You're going to have some leukocytes, some white blood cells, one with an undefined shape of the nucleus, one with a dark, large, rounded nucleus that occupies most of the size of the cell. So we call the one with undefined shape of a nucleus, this is going to be the neutrophil, and the one with a dark, rounded, large nucleus that occupies most of the size of the cell, this is going to be my lymphocyte. Let's see the type of connective tissue that is occupied, most of its size is occupied by fat cells, large vacuolated cells. Those vacuoles were compressing the nucleus towards the periphery of the cell. This is what gives the signet ring appearance to the cells. Those are my fat cells, and this is going to be my adipose connective tissue. See on here, another type of connective tissue that has a very characteristic appearance. It's formed of building blocks, and again, we call those building blocks are my osteons. Each osteon has a central canal in which we've got the blood vessels traveling, and surrounding the central canal, you're going to have layers of calcified protein. Those are my lamellae. Embedded in those lamellae, we've got those tiny little spaces where the bone cells, the mature bone cells, are going to be kept. Those are my lacunae, and the mature bone cells are called my osteocytes. So we're looking here at the osteocytes in lichen. Again, what type of tissue is this? This is my bone or osseous tissue. Bone or osseous tissue. Any question? Any question? All right, for the recording, I will try my best to post it for you. I will try my best to post it for you. All right, somebody was asking about video recording for this session. All right, so I will try to my best to post it for you. All right. All right, so let's have a break for 5. We'll come back and discuss the 2 other types of connective tissue, the connective tissue proper and the cartilage. All right. All right, let's have a break for 5. We come back, complete the discussion of the connective tissues. And we'll see you in 5. All right, welcome back. So just want to double check if you can still hear me and if you can still see the blackboard screen on your the blackboard on your screens. All right, so if we're looking at the other types of connective tissue, we have the cartilage. And the cartilage is so easy to identify by looking at cells that are separated from the surrounding matrix by a clear space. We call the cells of the cartilage are my chondrocytes, and the space surrounding those chondrocytes are going to be my lacunae. So if I see chondrocytes in lacunae, this tells me that I'm looking at a cartilage tissue. We have three types of cartilage tissue. We can differentiate them from one another by the appearance of the surrounding matrix. So all of them gonna share the same characteristic, which is the presence of chondrocytes in lacunae. If the chondrocytes are surrounding by a matrix that has a homogeneous consistency, I have almost the same color all around. I don't see much fibers in the matrix. So this tells me that I'm looking at a hyaline cartilage. If the surrounding matrix do show clear fibers, which are thin, darkly stained long fibers, so those are my elastic fibers, and I'm looking here at a type of cartilage which contains elastic fibers, so we're going to call it an elastic cartilage. If the surrounding matrix for the chondrocytes are, instead of showing those thin darkly stained long fibers, or they are not, it's not showing any fibers, it's showing thick bundles of fibers. And how would I know whether the fibers are thick or thin? Please compare the diameter of the fiber to the diameter of the cell. If I have larger diameters than the cell, it's definitely going to be a thick fiber, and those are going to be my collagen fibers on here. If the fiber thickness is much thinner than the diameter of the cells, so this is going to be an elastic cartilage, an elastic fiber, and this tissue is going to be my elastic cartilage. So here I've got a cartilage with extra fibers in it. So I'm dealing with a cartilage with extra fibers, so we're gonna call it fibrocartilage. Hyaline cartilage is gonna be present in the nose, in the larynx, in the tracheal rings, is gonna be also present connecting the sternum to the ribs, because those are my costal cartilages. It's also gonna be covering the articular surfaces of the articulating bones in a type of joints that we're going to be studying later called the synovial joints. So the synovial joints, which are representing the most common type of joints that we do have in the body, all the bony surfaces in the joints are covered with hyaline cartilage. Because cartilage is avascular, like the epithelium. Cartilage is avascular, so it doesn't get easily inflamed. That's why I will have them covering the articular surfaces to reduce the risk for inflammation because of the recurrent irritations that I have for my articular surfaces. Every time I'm moving, I'm applying friction on those articular surfaces, so I don't want to get them inflamed, so I will be covering them with hyaline cartilage, which is avascular. Similarly, you're going to have Similarly is going to be the other types of cartilage. So all types of cartilages are going to be avascular. Also forming the fetal skeleton that will become the adult skeleton with the bones. But before those bones become bones, they were actually hyaline cartilage. Elastic cartilage is going to be given flexibility to the organ it forms, and we need this flexibility in the ear pinna or the external ear, and we need this flexibility in a cartilage that is located above my larynx and my airways to prevent the food and fluids from passing down into my airways. We call this is my epiglottis. The fibrocartilage will allow you to absorb the compressive shock. Applying compression on this cartilage will be absorbed or partially absorbed due to the presence of the extra fibers in its structure. The fibrocartilage is going to be present between the vertebrae in the intervertebral discs, Also, it's going to be located in the menisci of the knee. So in the knee joint, I do have hyaline cartilage. I do have hyaline cartilage covering the articular surfaces, but I have extra fibrocartilage rings, which are going to be present also forming the medial and lateral menisci of the knee. So with every movement, when I'm applying compression on my spine, this compression is going to be partially absorbed by the fibrocartilage present in the intervertebral discs. Also, when I'm moving, when I'm running, I'm applying lots of compression here on my knee. So this compression is going to be partially absorbed by those menisci formed of fibrocartilage. Moving to the connective tissue proper. Connective tissue proper is subclassified into two major groups: loose connective tissue and dense connective tissue. And this is going to be determined according to the proximity of the fibers between so proximity of the fibers. So I need to look at the spaces between those fibers. I need to look at the proximity of the fibers to one another, whether or not I do have spaces between those fibers. So if I'm looking at a type of connective tissue that has collagen fibers in it, elastic fibers, and it has lots of spaces that contain scattered cells, like this, fibroblasts. I'm looking here at the areolar connective tissue. And remember, the areolar connective tissue is widely distributed in your body underneath the epithelium to allow the movement of the fluids and nutrients up towards your avascular epithelium. A second type of loose connective tissue, remember, this is the ones that we've stated earlier because we don't see much fibers in it. This is my adipose connective tissue. And why is it loose? Because you simply have the fibers far away from one another, and most of the tissue is occupied by the cells. So the fibers are far from one another. So that's why we classify the adipose connective tissue as a loose connective tissue. A third type of loose connective tissues was the one that contained the short branching fibers. Those are my reticular fibers. And remember, those reticular fibers are going to be surrounded by a large number of blood cells. Those are my precursor blood cells. We call this tissue that has reticular fibers, we call it the reticular connective tissue. And the reticular connective tissue, if you remember, it contains lots of blood cells scattered between its fibers, because simply it's going to be located within the organs that contain lots of blood cells. And those are my lymphoid organs. Like what lymphoid organs that we know? Like the lymph nodes, like the bone marrow, like this plane. And the function here is going to be to form the internal skeleton of those lymphoid organs. The second class of the connective tissue proper is going to be the dense connective tissue, in which the fibers are going to be closely arranged or closely packed against one another. So if I see that I have collagen fibers, and those collagen fibers are closely packed against one another, so this is going to be either a dense regular connective tissue or a dense irregular connective tissue. What differentiates the two from one another? Both of them are formed of collagen fibers. Both of them are going to be squeezing the fibroblasts in between those fibers. But the regular dense connective tissue is going to have a pattern of arrangement to its collagen fibers. Unlike the irregular dense connective tissue, you're going to have collagen fibers which are traveling in various directions. There is no clear pattern of arrangement of those fibers. They still have fibroblasts scattered and squeezed between the closely packed collagen fibers on here. No clear pattern of arrangement on here, so this is going to be the dense, irregular connective tissue. So we can see on here, you can see collagen fibers, elastic fibers, and lots of spaces in between those fibers. So this is going to be a type of loose connective tissue. What type of loose connective tissue has collagen fibers, elastic fibers, and the fibroblasts scattered around in those large spaces between the fibers? This is going to be my areolar connective tissue. If we're looking on here, I see those short branching fibers and huge number of blood cells scattered around those fibers. This is going to be my reticular connective tissue. And this reticular connective tissue is going to be present again in my lymphoid organs. Looking at this type of tissue on here, we can see definitely thick bundles of fibers. Those are my collagen fibers. Those collagen fibers are closely packed against one another. I don't have much spaces between the fibers, which actually cause the cells to be squeezed between the fibers. The fibroblasts here are squeezed between the adjacent collagen fibers. This is a dense connective tissue, but remember, if I see collagen fibers in a dense connective tissue, this might be either dense regular or dense irregular. If I see, as you see on here, the fibers are following a clear, regular pattern of arrangement here, like smooth waves. All right, so this is going to be a dense, regular connective tissue. If I see here no spaces between the fibers, those are definitely thick fibers because I see they are much larger than the diameter. They have a diameter much larger than the diameter of the adjacent cells. So those are collagen fibers. So I'm asking myself, do I follow any clear pattern of arrangement for those fibers or not? And the answer here is going to be that I'm not following any regular pattern of arrangement, so this is going to be classified as dense irregular connective tissue. In terms of function, the dense regular connective tissue allows you to apply tension in one direction. And you're going to need this functionality in your tendons and ligaments. What is the difference between a tendon and the ligament? The tendon is the one that connects a muscle to a bone, compared to a ligament, it's the one that connects two bones to one another. Unlike the dense irregular connective tissue, it's going to be able to accommodate and allow tension in many directions. And this is going to be present in the organ capsules. So whenever you're running, your liver is going to be moving up, down, sideways. So what keeps it in its place is going to be the organ capsule that is attached to the surroundings. So you're going to be always applying tension on those organ capsules. So why you've never heard of somebody rupturing the organ capsule? Because simply those organ capsules are formed by dense irregular connective tissue, which can tolerate tension exerted in many directions. This also includes the joint capsules. So you can move the joint in many directions without rupturing the capsule of the joint, but you might actually tearing the tendons and ligaments that are related to the joint. So although a person will not have a rupture of the joint capsule, but he might end up having a torn tendon or a torn ligament. Looking at another type of dense connective tissue in which I don't have collagen fibers, but instead I have those thin, darkly stained long fibers that are arranged very close to one another. Those are my elastic fibers. So when I see lots of elastic fibers closely packed against one another, this is going to be my elastic connective tissue, which is a type of dense connective tissue. So elastic connective tissue on here, as you see, elastic connective tissue, its main function is to allow stretch and passive recoil. So allows me to stretch and passively recoil. We're going to need this functionality in the large arteries, as we've discussed, to maintain a pulsatile blood flow. So during cardiac contraction, the artery is going to be expanding, stretched, and when the heart is relaxing, the artery is going to recoil, pushing the blood forwards. So what are the different possible answers that you can have on your exam? This is very important to know. So can you tell me what possible answers can I have for epithelium? For identification questions, not function, not location. So for the epithelium, we can have simple squamous, simple cuboidal, simple columnar, either ciliated or simple columnar non-ciliated. We can have pseudo-stratified, columnar, either ciliated, and very unlikely pseudo-stratified columnar non-ciliated. This is very unlikely. We can have keratinized, stratified squamous epithelium. We can have non-keratinized, stratified squamous epithelium. And we can have transitional epithelium. What are the possible connective tissues that you can see? I can see blood, bone, or osseous tissue. I can see hyaline cartilage, elastic cartilage, fibrocartilage. I can see areolar connective tissue, adipose connective tissue, reticular, dense regular, dense irregular, and elastic connective tissue. Also, I can see nervous tissue, I can see muscle tissue that either can be skeletal muscle tissue cardiac muscle tissue, or smooth muscle tissue. So on the exam, how many choice would I have to pick from the correct answer? You will be choosing between 9 and 11, 20, and 4. So you have 24 possible answers for identifying the tissue. So when I study, I need first is it kind of set up like the lab, but with more answers? Yes, exactly. So you're going to have a list of choices like the lab, but you're going to have all possible choices. And actually, you might have something which is totally wrong, like something like stratified that we didn't even study, stratified cuboidal or stratified columnar. All right, so you might even have more than 24 possible choices. It's the same list you're gonna be picking the single best answer from for each question. The same will apply for locations and functions. So when I'm preparing for this exam, what I need to do is to make sure that I'm able to identify those different tissues. Start first with the ones that you have in your slides, the regular slides that we have been working from the PowerPoints, and then move on to the ones that you have in your lab assignment on visible body. So for you to be familiar with both sets of slides. So make sure you are able to identify the tissue first. It's very important. For locations and functions, what I highly recommend you is to create this list, recreate this list on here. Build a table, for example, and try to test whether you are able to recall all different locations and all different functions of each of those tissues or not. And mark down the ones that you did not recall and check them. And then keep repeating this process until you are comfortable recalling all locations, all possible locations, all the functions that are stated here on the PowerPoint presentation. All right, because on the exam, I might be giving you, let's say, the function of or location of simple squamous epithelium. I give you this example back in class. So simple squamous, let's say. So I'm asking about location of the simple squamous. You have a long list of choices. And the good answer is the glomerulus, for example, or the glomeruli of the kidney. If you did just study one location for each of those tissues, and it was not the glomerulus, you wouldn't figure out the good answer on the test. So that's why you need to know all different locations for each of those tissues. Yes, the only thing that will be allowed on the exam is going to be a scratch paper. The only thing that's going to be allowed on the exam is going to be a scratch paper.